to present you the cloning.
When you hear the word cloning, you may think of the cloning of whole organisms, such as only the sheep. However, all it means to clone something is to make a genetically exact copy of it. In a molecular biology lab, what's the most often cloned is a gene or other small piece of DNA. DNA cloning is a molecular biology technique that makes many identical copies of a piece of DNA. In a typical DNA cloning procedure, the gene or other DNA fragment of interest is first inserted into a circular piece of DNA called plasmid. The insertion is done using enzymes that cut and paste the DNA, and it produces a molecule of recombinant DNA. Next, the recombinant plasmid are selected and grown up. As they reproduce, they replicate the plasmid and pass it on to their offspring, making copies of the DNA it contains.
How can pieces of DNA from different sources be joined together? We can use the restriction enzymes, and DNA ligase is a DNA cutting enzyme that recognizes a specific target sequence and cuts DNA into two pieces that are near that site. Many restriction enzymes produce cut ends with short single-stranded helix. Plasmids and other DNA can be introduced into bacteria in a process called transformation. During transformation, specially prepared bacterial cells are given a shock, such as high temperature, that encourages them to take up foreign DNA. A plasmid typically contains an antibiotic-resistant gene, which allows bacteria to survive in the presence of a specific antibiotic.
Bacteria without a plasmid will die, while bacteria carrying a plasmid can live and reproduce. Not all colonies will necessarily contain the right plasmid. That's because during a ligation, DNA fragments don't always get pasted in exactly the way we intend. We can grow a large culture of plasmid-bearing bacteria. Then we give the bacteria a chemical signal that instructs them to make the target protein. If our plasmid contained the human insulin gene, the bacteria will start transcribing the gene and translating the mRNA to produce many molecules of human insulin protein.
DNA moleculars are used for many purposes in molecular biology, like biopharmaceuticals. DNA cloning can be used to make human proteins with biomedical applications and human growth hormone. Gene therapy. Gene therapy attempts to provide a normal copy of the gene to the cells of a patient's body. When the plasmids were delivered to the lungs of cystic fibrosis patients, lung function deteriorated less quickly. Gene analysis. Biologists often use DNA to build artificial versions of genes that help them to understand how normal genes in an organism function.
Now we will present you a list of animals that have been cloned in the past. This list is subheaded by animals of different types. The first animal I'm going to speak about is a camel. It was called Injaz, and it was a cloned female dromedary camel. It was born in 2009 at the Camel Reproduction Center in Dubai after an uncomplicated gestation of 378 days.
Embryologist Tom Ditsu successfully inserted the DNA from a male Asian carp into the egg of a female Asian carp to create the first fish clone in 1963. In 1973, Ditsu inserted Asian carp DNA into European crucian carp to create the first interspecies of this clone.
In 2001, scientists at Texas University created the first cloned cat. Even though the copycat is an exact copy of her host, they have different personalities. The copycat is shy and timid, her host, on the other hand, is playful and curious. In 2004, the first commercially cloned cat, Little Niggy, was created.
Arctic wolf was cloned by South Korean science, including the controversial science Wang Wusak in 2005. The two female cloned wolves were housed in a zoo in South Korea for public view. The wolves were called Snowwolfie and Snowwolf, which were names taken from Seoul National University. Snowwolf died in 2009 from an infection.
Possibly the first cloned mammal was a sheep by Steen Villatzen in 1984. However, the cloning was done from early embryo cells, while the sheep dolly in 1996 was cloned from an adult cell. Megan and Morag were sheep cloned from differentiated embryonic cells in 1995. Dolly, the first mammal from adult somatic cells, had 6 lambs. In this experiment, we will use banana, absolute ethanol, water, liquid soap, paper filter, magnifying glass, test tube, plastic dishes, plastic spoon and knife, and a syringe.
Of salt, about 10 grams in 100 milliliters of water.
Add a spoonful of liquid soap.
Mix slowly without making foam, which could hinder the contact between the liquid and the banana.
Take half a banana and crush it.
Add the banana pulp to the solution and wait for at least 20 minutes.
Filter the mixture.
Take 5 milliliters with a syringe.
Transfer the liquid to a test tube and try not to foam.
Invert the tube 2 or 3 times to promote the mixing of liquids.
Add 10 milliliters of alcohol for food.
A few minutes for the ball of DNA to get into the alcohol.
In the past, cloning has been criticized for ethical reasons.
In our opinion, cloning will be very useful for humanity. It could improve the surgery field, avoiding the transplant rejection.